All right, this slide range is all about mutation. And there are two different kinds of mutations. But in general, a mutation is any change in the DNA nucleotide sequence. Any change at all. One letter, that's a mutation. One nucleotide is what I should say. It's caused by mistakes during DNA replication, and you know all about DNA replication, as well as mutagens, like certain chemicals and radiation. So, for example, Teenage Mutant Ninja Turtles, as the story goes, these little baby turtles accidentally got down into the sewer where they came in contact with a chemical mutagen that turned them into the Teenage Mutant Ninja Turtles. What does not happen is what I just described. The fictional misconception that a mutagen can transform an individual into a monster or a superhero. That doesn't happen. Individuals, just like individuals don't evolve, individuals cannot be turned into a completely different organism because of a mutagen. It can cause cancer, however, and when a mutagen causes cancer, we call that a carcinogen. So mutagens can be carcinogens and cause cancer, cause mutations in body cells that then go rogue and multiply uncontrollably, and that's cancer. Gene mutations occur during DNA replication, so we're talking specifically about gene mutations here, when the nucleotides change. And that happens before cell division, before cells divide, and from damage caused by mutagens, of course. For example, ultraviolet radiation will cause changes in the nucleotide sequence, in nucleotides. Chromosomal mutations happen both during replication and cell division. Mostly cell division, though. So we're talking about chromosomal mutations here, where whole pieces of chromosomes are changed. Gene mutations usually affect the gene, one gene at a time. So, for example, just changing one nucleotide is only going to happen in one gene. Genes can be thousands of nucleotides long, and one change in those thousands of nucleotides may or may not make a difference. But that's typically, gene mutations are affecting a very small portion of the DNA, whereas chromosomal mutations can affect hundreds to thousands of genes at a time and are usually lethal. Usually, someone who has a chromosomal mutation will not even be born. And a lot of miscarriages, or also known as spontaneous abortions, are probably the result of chromosomal mutations. So we'll start looking at gene mutations first, and this first one first of all, let's list the different kinds of gene mutations that there are. And what we're talking about here are referred to as point mutations because they affect just one nucleotide in the DNA nucleotide sequence. So that's just one point in the gene. One nucleotide, one point. So we call it a point mutation. It may affect one or two, but in general, we're talking about just a very small change in the DNA nucleotide sequence. And that can involve substitution, where one nucleotide is substituted for another. Insertion, where a nucleotide is squeezed in between two nucleotides. And deletion, where a nucleotide is completely eliminated. What we're looking at here is substitution. So notice that in the DNA here, and notice what we're looking at here, the whole process is the central dogma of biology. It's most of gene expression. So we start with DNA that's transcribed into the RNA, and our original DNA has a C here in this position that's transcribed into a G. Our mutation, though, replaces the C with a T. And that changes the nucleotide and messenger RNA to an A, which completely changes the codon, right? So we go from a codon that is CGU, and if you look that up on the circular table, the genetic code table, that codes for arginine. But after the mutation, the codon is CAU, which codes for histidine. And this is all just describing that. Arginine to histidine. And this may or may not affect the final conformation or function of the polypeptide or protein that we're dealing with here. We're talking about most polypeptides are hundreds or even thousands of nucleotides, of amino acids long. And one change may or may not make a difference in the function of that protein. And it can go a number of different ways. It can be helpful. It can actually make that protein function better. And if you think about that from an evolutionary standpoint, then that's possibly going to be selected for by natural selection. And therefore it's going to be maintained in the population. So, this is where the differences between individuals, the variation between individuals ultimately begins, is with mutation. And sometimes those mutations are helpful, sometimes they're good, and they're selected for by natural selection. They can also be neutral, and natural selection won't act on them at all because they're neutral. They don't make any difference at all. So if this change from arginine to histidine makes absolutely no difference in the function of the protein, then it can stay, and it can be passed on to offspring, but it's not really going to be selected for or selected against. It's neutral. If it's harmful, if it does make that protein function decrease the function of that protein, then it's going to be selected against, or it could be selected against. Depends how severe that reduction in function is. And if it's really severe, then it could be lethal. It may eliminate the function of the enzyme or some other protein that's crucial to survival, and therefore that person would never be born, or would die shortly after being born, or at least not live long enough to reproduce. So in other words, selected against by natural selection. Another kind of point mutation is insertion, and that's what we're looking at in this scenario. And in this case, the T there's an insertion of a T between the A and the C. So here's our original nucleotide sequence in the DNA. Notice that we're squeezing a T in between the A and the C, and that's causing the C to move over, shift, as we say, downstream in this case. And it not only shifts that C, it shifts everything after the C. And where that's a problem is when it's transcribed into RNA, because now the RNA codons are going to be read differently. So this is just describing what's going on here. So what we call that is a frame shift, where insertion causes a shift in all the nucleotides downstream from where the insertion took place, changing the reading frames, as we say. That's why it's called a frame shift. It changes the reading frames of the codons so that now all the codons from that point on are going to be read differently. And that may or may not make a difference. Notice, too, that in both this and the example on the previous slide, the very first codon was AUG, which codes for methionine, which is the start codon that all proteins start with. Well, check out what's happening in this scenario with this insertion. The change in that nucleotide where the insertion took place is from AUG to AUA, which changes the methionine to isoleucine. And isoleucine is not the start amino acid. Therefore, this protein, this polypeptide chain, would never be even made. So it's catastrophic. This insertion mutation is catastrophic to whatever this protein is, because it would never be made. There is no start codon now. So it would not, initiation of protein synthesis or translation wouldn't even happen. So this frame shifts result in most of the amino acids being wrong. Notice that leucine here is the same. Leucine is still in the same position, and it's still leucine, but that's the only one that hasn't changed. So usually these are catastrophic. They're potentially the most severe, most often completely eliminating the structure and function of the resulting polypeptide or protein. Frame shifts can also result from deletion, and in this case, in this example, we're looking at a sentence that's made up of 3-letter words to represent to be the same thing as the 3-letter codons in messenger RNA. So the fat cat ate the rat. The fat cat ate the rat. You know, simple sentence, 3-letter words. But if we eliminate the H in the, then the reading frames are shifted. And if we read those letters in 3-letter words starting from the beginning, it's nonsense. It doesn't make any sense. It's missense, as we say in genetics anyway. This is just describing what we just went through. So just as the sentence no longer makes sense, neither would the messenger RNA nucleotide sequence. A protein resulting from this deletion and frame shift would have a completely different structure, eliminating its function. You know, this protein would not function at all because none of the codons are the same. So we identify mutations as either being missense or nonsense. A missense mutation is like we see here. There would be amino acids produced and strung together as long as this isn't a change in the start codon. But it looks like it is because that mutation is in the very first word, which is representing the first codon, so it would be. But if it doesn't affect the start codon, then it would be missense, and simply all the amino acids that go into that protein, or nearly all, would be changed, and we call that missense. So the protein would be produced, the polypeptide chain would be produced, but it would have the wrong amino acids. Nonsense is when there's either no start codon or there's a stop codon inserted somewhere in that polypeptide so that only part of the polypeptide is made. So maybe half of the polypeptide is made, and that would be catastrophic to the structure and function of that polypeptide. So it depends on where that stop codon would be inserted. If it's inserted way downstream, almost near the end of the polypeptide, it may not even make a difference. It could be a neutral mutation. But in general, frame shifts that result from either insertions or deletions are the most catastrophic kind of gene mutation. So gene mutations is one kind of mutation. The other kind of mutation are chromosomal mutations that affect whole chromosomes. And we haven't looked at the structure of chromosomes very much yet, just a little bit, but you have 46 chromosomes. And what can change about the chromosomes is their structure or their number. And either one of these can produce what we usually refer to as syndromes. But a lot of times chromosomal mutations are fatal because they, like I mentioned earlier, affect whole pieces of chromosomes. Like, for example, this B section of this chromosome can have hundreds or even thousands of genes. And if something happens to that section, it affects all of those genes. So this diagram represents chromosomal mutations of structure. Just like in deletion in a gene mutation, deletion is simply the loss of not just one nucleotide in this case, but a whole section of the chromosome. So notice as we go from the original chromosome here to the deleted example, we no longer have section B. It's gone. And again, that's hundreds or thousands of genes that have just simply been lost. And that can be catastrophic, usually is catastrophic. Or it's going to produce some kind of syndrome. There's also duplication. And that's going to happen during DNA replication. So if we go from the original chromosome again to this one representing duplication, we've got now two sections of section B. And that can and that may or may not make a difference. When you have more genes, that means more transcription and translation into protein, and that means more protein. And you might think to yourself, well, what can be so bad about having extra protein, double the protein? Well, that can cause a problem, because the amount of protein in your body, think of enzymes, the amount of enzymes in your body has to be pretty tightly regulated so that their activity is not too much. Too much enzyme activity can cause just as many problems as too little enzyme activity. So this can be a problem, or it may not. Inversion is when a whole section of chromosome flips. So notice what's happened here is, if you read this backwards now, it's B, C, D, E backwards. So this whole center section of the chromosome has flipped. And that can cause changes or breaks in genes. So for example, like I note here, where it flips, where it breaks to create the flip, can break genes. So there can be a gene right here that now is broken and therefore it won't function. Same over here. A gene is broken and possibly broken and won't function. But some genes are together on chromosomes and need to be read in order. So this will change the order that genes are going to be expressed. So it separates genes so that they won't be expressed together. And that can cause problems. And then finally, we can have translocation, where a piece of one chromosome breaks off and moves to another chromosome. So here we see JKL, that's not even in our original chromosome, that came from another chromosome. And we can have reciprocal translocations. So for example, the chromosome that JKL came from may have received DEF from our original chromosome. So they swapped pieces. They switched pieces. And again, that may or may not cause a problem. That may be completely neutral, but it might separate genes that are supposed to be read together, and it might break genes so that they don't function. So chromosomal mutations are often fatal because they affect many genes at once. Most severe is deletion, because genes are lost. So in these other scenarios, the genes are still there, they're just in a different arrangement. But deletion, the genes are gone. And as I mentioned, overproduction of proteins is not necessarily a good thing. And also, as mentioned, inversion and translocations can break or separate and reorder genes, put them in the wrong order. So those are chromosomal mutations of structure. Now let's take a look at chromosomal mutations of number. So you'll notice that my daughter's name is here, Kayla Zagre, and what we're looking at here, notice it's labeled normal female karyotype. A karyotype is kind of like a picture of the chromosomes from an individual, but they are arranged in a specific order. So all of human chromosomes, all the 46 human chromosomes, they're in pairs, and excuse me, and they're numbered. So when a karyotype is produced for a person, their chromosomes are arranged in pairs and placed into order, this very same order. So we can talk about human chromosome number 2, and we can talk about human chromosome 21, and so forth. And everybody knows what chromosome we're talking about. So this was done before my daughter was born by a process known as amniocentesis, where they take a long needle and they put it through the mother's abdomen, into the womb, into the uterus, and into the amniotic sac, or the amniotic membrane that's surrounding the baby, and they draw out amniotic fluid that the baby is floating in. And in that amniotic fluid are cells from the baby. You know, just like your skin is constantly sloughing off cells, and your cheek epithelium is constantly sloughing off cells, for example, and we're going to be looking at that under the microscope in the next lab, by the way. But anyway, cells from the amniotic fluid can be obtained from the baby, and they look for cells that are undergoing mitosis, in other words, cell division, because those are the cells that have fully formed mitotic chromosomes. That's what we're looking at here are mitotic chromosomes. Chromosomes that have condensed in preparation for cell division. And when they find one of those cells, they can break it open and spill out the chromosomes. So this inset photo here is a picture of that, where they've broken open a mitotic cell, a cell undergoing cell division, or preparing to undergo cell division, and they've spilled out all the chromosomes. And then they take a picture of it. So these chromosomes that are in the karyotype are the same chromosomes that are in this picture. It's just that they've been cut out of the picture and placed in order, in pairs and in order. Notice that the order is basically according to size and banding pattern until we get to the end, because when we get to the end, these are the sex chromosomes. And the X chromosomes are big. Y chromosomes are really little, but you'll notice that there is no Y chromosome here because my daughter's my daughter and not my son. 2 X chromosomes makes her a female, right? So this is normal female karyotype. So anyway, this represents chromosomal mutations of number, and the name for that, the general name for any chromosomal mutation of number, any wrong number of chromosomes, is known as aneuploidy. So that's the general term. Polyploidy is a term that refers to having extra chromosomes. So the wrong number of chromosomes, too many chromosomes. So for example, strawberries, and a lot of our crop species that we grow for food is polyploid. And they've been created polyploid by by selective breeding, but sometimes inbreeding, and also causing mutations, causing non-separation of chromosomes during cell division. In that way, they can produce polyploid species. And that's why strawberries grow so big, because they're polyploid. The little wild strawberries that are growing in my backyard, for example, are teeny compared to the ones that we grow and eat. And there have been strawberry species and hybrids and varieties that have anywhere from 2 to 4 to up to 10 sets of chromosomes. So that would be like a human having, double, triple, quadruple the number, of 46 chromosomes. So like I said, this is a karyotype. I've already described that. So it's basically the chromosomes from an individual arranged into pairs and in order according to size and banding pattern. But we can look for chromosomal mutations of both structure and number from this. So it can diagnose things like Down syndrome, which would be an extra chromosome 21. And another syndrome that you may or may not have heard of, known as cri du chat, which means cry of the cat in French. And that's because those that have this syndrome have a very high voice, and when they're babies, they sound like a crying cat. And that's due to a missing piece of chromosome number 5, I believe. And again, this is just these last 2 points are things that we've already looked at. So there can be neutral mutations. So you can have a mutation of chromosome number or structure, or a point mutation in a gene mutation that is neutral and makes no difference. And I guarantee you, we all have mutations. Every single person on this planet has a mutation, most likely neutral, at least one, if not many more. So mutation is actually beneficial from a big picture point of view, from the perspective of natural selection and evolution. It's where variation comes from. It's the beginning of variation. Every difference between every individual and every species on this planet started out as a mutation. And we'll leave it right there.